Hello everyone, today we will discuss the part 16 for important notes for biochemistry so that you can easily able to answer the one-liners and the MCQs in your exam. First of all, the enzymes are actually high molecular weight, highly efficient and highly specialized protein which catalyze the biochemical reactions in the body. Enzymes actually increase the rate of reaction without making any change in the overall process. All enzymes are proteins except for the ribozyme. Enzymes are substrate specific, stereospecific and thermolabile. Active site of enzymes is made up of, remember, mainly two sites, that is the binding site, usually 2 in number, and catalytic site. Okay, so as you can see over here, this is the enzyme, this yellow thing is the enzyme, okay, and this is the active site of the enzyme, which is actually having two sites, that is the binding site and the catalytic site. Active site is flexible and not rigid. The active site of enzyme is form of amino acid which may be placed at long distances on the primary structure but are brought nearer to each other by the three-dimensional conformation of the enzyme. Induced fit theory. Now what is this induced fit theory? The active site of a particular enzyme is flexible. When substrate approaches the active site, as you can see over this, the active site. So whenever the substrate actually approaches the active site, then there is conformational changes occurring, now which actually induces the active site to fit the substrate. As you can see, the enzyme actually changes its shape slightly as the substrate binds. Okay, and there is conformational changes occurring which induces the active site to fit into the substrate. For any thermodynamically favorable reaction, the free energy of the product is lower than that of the substrate. The amount of Gibbs free energy tells that a reaction is thermodynamically favorable or not. So, if the Gibbs free energy is actually less than 0, means if the Gibbs free energy is negative, then the particular reaction is thermodynamically stable. If the Gibbs free energy is positive, or we can say more than 0, then the reaction is thermodynamically unfavorable, or we can say unstable. And if the Gibbs free energy is equal to 0, then the reaction is in the equilibrium condition. The enzyme decreases the activation energy as well as the time required for the reaction. As you can see in this graph, okay, this graph is with enzyme and this is without enzyme. Okay, so as you can see, the activation energy of this particular reaction actually reduced with enzyme, even the time reduced, right? Enzyme does not affect the free energy or the equilibrium of the reaction. Holoenzyme. Now what is the holoenzyme, or we can say the active enzyme? So apoenzyme, or we can say the protein portion, this is the apoenzyme or the protein part plus non-protein part. Okay, now this protein and the non-protein part which we call as cofactor. Okay, now these two actually combines and forms the holoenzyme. Okay, now apoenzyme, that is this portion, this is the protein portion. This apoenzyme alone is inactive, but when this apoenzyme combines with the non-protein portion, this both actually combines and forms the active holoenzyme. Okay, but alone this apoenzyme is inactive. Now what about this non-protein portion? Now this non-protein part is of two types. It can be cofactor or it can be prosthetic group. Now if the non-protein portion is separable or dialysable from the protein portion, then it is known as cofactor. Okay, now if this particular cofactor is organic, then it is known as coenzyme. Okay, so if the non-protein portion is separable or dialysable from the protein portion, then it is known as cofactor or coenzyme. Okay, if it is actually organic, then coenzyme. And if the non-protein portion is not separable from the, or we can say is actually non-dialysable from the protein portion, then it is known as prosthetic group. Then this non-protein portion is known as the prosthetic group. Okay, and this apoenzyme and the cofactor, or we can say coenzyme, or we can say prosthetic group, together forms the holoenzyme. Mostly metal is prosthetic group. Enzymes which require metal ion as cofactor are known as metal activated enzymes. Enzymes which require metal ion as prosthetic group are known as metalloenzyme. 1/3 of the human enzymes are metalloenzymes. All coenzymes are cofactors. Metals are cofactors and not coenzymes. Coenzymes may be covalently or non-covalently linked. Coenzyme Q and glutathione acts as coenzyme. Some metal enzymes contain more than one metal. For example, cytochrome oxidase contains both iron and copper. Coenzyme act as hydrogen carrier as well as carrier of moieties other than the hydrogen. SAM, that is the S-adenosyl methionine, it is actually a methyl donor. PAPS, that is the phosphoadenosyl phosphosulfate. Now this PAPS, phosphoadenosyl phosphosulfate, over here, this PAPS are actually sulfate donor in synthesis of GAGs, or we can say glycosaminoglycans, and synthesis of sulfoglycosphingolipids, and in the steroid hormone catabolism, this PAPS actually acts as the sulfate donor. SAM and PAPS, that means the S-adenosyl methionine and the phosphoadenosyl phosphosulfate, are nucleotides but not coenzyme. Vitamin K is the only fat-soluble vitamin which acts as coenzyme. Lipoic acid is actually a coenzyme but not a vitamin. All water-soluble vitamins act as coenzymes. All oxidases need copper except for the xanthine oxidase and sulfite oxidase, which require the molybdenum. All kinases need magnesium, but pyruvate kinase need potassium more than magnesium. All carboxylases need magnesium. Cytoplasmic superoxide dismutase require copper, but remember mitochondrial superoxide dismutase require manganese. So today up till here, we'll discuss about the further important notes in our next video. Thank you so much.